This is a video set on the origin of cells. So one thing I want to stress in here is that you don't need to remember years, you need to remember the big moments that we're going to highlight and summarize in here. But it's worth knowing that the Earth is 4.6 billion years old. That first environment was very volatile, so meteors regularly hit the Earth's surface. There was a lot of radioactive decay, lighter elements were floating to the surface, and volcanoes were releasing a lot of gases that were trapped inside the Earth's crust. So lots of carbon monoxide, which is poisonous to us, carbon dioxide, water vapor, nitrogen, and methane gases were in the atmosphere. It's not like it was today. As you noticed on the last list, there definitely was no oxygen, and there was also no ozone, which is O3. So UV rays were very, very harmful because there was no ozone layer to protect any organisms or anything on the surface of the Earth from those UV rays from the sun. There was also no greenhouse effect, which made the environment extremely cold. The greenhouse effect is a warming effect where it traps sunlight's energy and gases inside the Earth's atmosphere and warms it up. So again, really stressing that early atmosphere lacked oxygen. Cooling the atmosphere, so the atmosphere cooled and it created heavy rains from all that water vapor, and that led to shallow seas, which gave scientists the idea of the primordial soup as an initial model of where life began. And so scientists suggested that the Earth's oceans contain large amounts of organic molecules from the Earth's crust. It's believed that the atmosphere lacked oxygen again, but contained those gases such as nitrogen, hydrogen, methane, ammonia, and many other gases. But again, it lacked oxygen. Just super important that early atmosphere lacked oxygen. Scientists suggested that the chemicals and the simple molecules in that early shallow sea led to the building blocks of life. So those non-living matters reacted chemically to form simple molecules. If you notice, those should look familiar because those are our 4 organic compounds. So nucleic acids, amino acids, fatty acids, and hydrocarbons. Another term for hydrocarbons is glucose, right? So hydrocarbons are the building blocks for carbohydrates. Fatty acids are the building blocks for phospholipids. Amino acids are the building blocks for proteins, and nucleic acids are the building blocks for RNA and DNA. So it was suggested that these molecules came together to form what will become the 4 organic compounds and molecules. So primitive cells formed 4 billion years ago, and it was an RNA polypeptide loop surrounded by a lipid membrane. Scientists call this a protocell, and we'll get into further how they suggest this formed in just a little bit in these notes. Bacteria-like fossils, so our prokaryotes, formed about 3.5 billion years ago, but because there was no oxygen yet, these were anaerobic, so without oxygen. Bacteria. Oxygen was produced by cyanobacteria, so cyanobacteria is a huge player in the game because it was the first organism that started producing oxygen, and oxygen then accumulated in the atmosphere. As soon as that started happening, fossils started appearing from organisms that could use oxygen, and then more complex things such as eukaryotes started to appear. So once those anaerobic fossils appeared, again, anaerobic is without oxygen, so our anaerobic prokaryotes appeared. We then see that there was photosynthetic cyanobacteria, so you'll hear it called just cyanobacteria or photosynthetic cyanobacteria, and again, this importance is that it released oxygen into the atmosphere. Then we saw aerobic bacteria appear, so our aerobic prokaryotes. So we had anaerobic prokaryotes first, and then we had our photosynthetic cyanobacteria, and then we had aerobic prokaryotes. So aerobic means with oxygen, and then there's things in the environment now that can use oxygen, things in the soup, if you will. And then once we see that, eukaryotic fossils start to appear in the records. We see more protists found in this time. Protists are simple-celled eukaryotic organisms. More complex cells are then developing multicellular organisms, and then animals and plants phyla also start appearing within that evolutionary history. So cyanobacteria, it all started with cyanobacteria producing that oxygen in that early atmosphere. That way aerobic bacteria could appear, and things that use oxygen from the environment, organisms that use oxygen, were then appearing. So Stanley Miller tested the soup model to see if molecules could form spontaneously in chemical reactions, and he showed that organic molecules can form spontaneously, but this theory has now changed with the finding of deep ocean vents. So the current thought is that life started near the vents in the deep ocean. It is suggested that no UV rays and chemicals were available for energy consumption, which leads to our understanding of what are called archaebacteria. And these are bacteria that live in extreme environments. So these are extreme environments are thermophiles and halophiles. So it's suggested that life near those deep ocean vents were organisms that were anaerobic and that they made their own food from chemicals. Okay, again, bacteria that live in extreme environments are considered archaebacteria. We will build on this as well in the bacteria notes. Archaebacteria were thought to only live in extreme environments, but now we know that they live in pretty much any environment, but they also are the only type that live in extreme environments. So exactly where organic molecules started is still in the hypothesis stage, but that they did form is not a dispute. So from simple monomers, so monomers is, remember, mono one, so simple things to complex polymers surrounded by a membrane equals a protocell. And then next came RNA, so our nucleic acids all in a row, creating RNA that could replicate. And the idea of this is called a protocell. So this was kind of the first cell. This is a diagram of how those cells were formed. So it has been tested that if you have lipids mixed with macromolecules, such as proteins or nucleic acids, and they dry it and wet it, just like in a tide pool, the macromolecules are then readily captured. So that means that those lipids will kind of flatten out, and when they rehydrate, they're going to form that circle and capture nucleic acids and proteins on the inside of the membrane, and that's what we call membrane-bound vesicles, or that's probably the beginning start of the understanding of membrane-bound organelles. So this is just how the protocell is thought to have been created. We need to know a couple words in science, so you'll hear this a lot. So autotroph. Autotroph means self-feeder, so that's definitely a vocabulary word you need to know. And heterotroph means that it's an organism that must consume food for energy. So we are all heterotrophs. Plants are autotrophs. Okay, so again, two very important vocab words you need to know. So prokaryotic development, the first thing that came was a chemolithoautotroph. So this was using chemicals to make their own food, and these were anaerobic, so it's using sulfur and other chemicals. These are thought to be the first archaebacteria that were found in those deep ocean vents. Heterotrophic prokaryotes came next with an opportunity to eat those autotrophs, so these are still prokaryotes, still very simple cells. Photosynthesizing prokaryotes, so this is our cyanobacteria. So these guys are releasing oxygen into the atmosphere, and that created what we call the aerobic explosion. So aerobic means with oxygen. So as soon as those photosynthesizing prokaryotes showed up, we then had an aerobic explosion. Okay, an aerobic explosion is because it put oxygen into the atmosphere. So again, just our example of our prokaryotic cells so you don't forget. Simple structure, no membrane-bound organelles in the middle, but a membrane-bound cell in and of itself. Sometimes it has a cell wall, sometimes it has an endospore, and we'll get into that structure when we talk more about bacteria. So development of eukaryotes. Endosymbiosis is the theory that suggests that prokaryotes were ingested by larger cells, and those became our membrane-bound organelles. So it is suggested that mitochondria and chloroplast come from prokaryotes that used to use oxygen and create energy. So a single bacterial cell, and chloroplasts used carbon dioxide and sunlight to create energy. And a larger organism engulfed those two things. For example, here, infolding of the plasma membrane, and it's engulfing here these aerobic heterotrophic prokaryotes, so oxygen using, needs to eat other things, simple-celled organism here, and it's engulfed that, and it was a symbiotic relationship, which means that it worked together, so it helped each other. And that's where we talk about, when we get more into eukaryotic cells, is that a mitochondria has a double cell membrane, and it's because the outer, like the inner membrane here was its original prokaryotic membrane, and then the outer membrane is coming from when that first larger prokaryote engulfed it and kind of left its membrane on the outside. The same is suggested for chloroplasts here, engulfing of photosynthetic prokaryotes. So photosynthetic means it's going to use the sunlight to create energy, and that's where we're getting our plant cells. So here's an ancestral photosynthetic eukaryote. Notice our plant cells have mitochondria and chloroplasts. And then here's an example of an ancestral heterotrophic eukaryote, where we've got a little bit more complex cells because we've got membrane-bound organelles. And then we have just mitochondria in our heterotrophic eukaryotes, so suggesting that they had to continue to ingest other organisms and other chemicals to be able to continue to make energy. It's suggested here with this photosynthetic word that this organism was able to create its own energy because it engulfed the photosynthetic prokaryote. So that's the theory of endosymbiosis and how we started getting our membrane-bound organelles and creating those eukaryotic cells. So here's just a kind of look at from our protocell to our kingdoms here, so that's kind of interesting. So just a summary of how cells develop. The first is that we had simple organic materials, and then we had complex organic strands, so we created our polymers, and then we had our protocells, followed by our chemolithoautotrophs, and remember those would be anaerobic because there wasn't oxygen yet, and then we had our heterotrophic prokaryotes, and then we had our photosynthesizing prokaryotes. So remember, cyanobacteria is putting oxygen into the atmosphere. So O2 is oxygen, ATM is a shortened way to say atmosphere. Finally, we had eukaryotes, followed by more complex organisms. So there's our origin of life and summary of how cells developed.